Hello everyone, I'm Kunal. I'm a grad student at Institute for Bioscience and Biotech Research, which is part of University of Maryland, College Park. And this is my first time that I'll be interacting with a visualization group here. So I would really encourage to give feedback on my work that I'm going to present. The title of my talk, as you see, is MecCog, Framework for Representing Human Genetic Disease Mechanisms. So with the advancement in high throughput technologies, we are getting new insights about mechanisms connecting perturbed genotype to perturbed phenotype. Now what are perturbed genotypes? They are variants, genetic variants, GWAS identified SNPs or driver mutations in cancer. And perturbed phenotypes are monogenic diseases, complex trait diseases, or cancer. Now if you want to really read about these mechanisms, in PubMed or PubMed Central, they're pretty much scattered and they're represented very diversely. They could be either in form of, all right, do you see a pointer? Okay, either in form of schematic diagrams or in form of networks, sometimes in form of just free text in tables, and they are pretty much scattered across multiple publications. So that kind of creates a major challenge here, that how do we extract and organize and represent this knowledge to maximize its accessibility and utilization. So this calls for an idea that we need something to integrate and present this disease mechanism knowledge in a coherent, comprehensive, and intuitive way. So that was the kind of an idea which brought us to create this framework called MecCog, which is available in www.meccog.org, which is basically a diagrammatic framework for representing human genetic disease mechanisms in an expert source manner. And MecCog is built on the formal concept of biological mechanisms, which is a work from the field of philosophy of biology, which has been done by Craver and Darden. And this is a reference for their work. I don't know if you can see the name, but the name of the book is In Search of Mechanism. It's an interesting book. You might want to read it. And Professor Darden is part of our MecCog project. So what is this framework all about, and how does it look like? So this is mechanism connecting a perturbation to a perturbed phenotype. Now you can break a mechanism into multiple steps, and these steps are steps of triplets. Something like this, which is a steps of triplets of SSP, MM, SSP, where SSP stands for entity. For instance, it could be a single nucleotide variation or an indel. And MM stands for activities and entities that can alter these entities. And they could be transcription, translation, etc. And you can imagine these MMs as connecting, as edge labels connecting two SSPs, where SSPs could be nodes in a network, and the MMs are basically labeling the edge that connects these two. And as an example, here's a SNV, which is an input to a transcription mechanism module, which causes a change in RNA, which is an input to a translation mechanism, which changes the protein, and so on and so forth. And you can imagine this kind of a structure connecting a SNV to a disease phenotype. And such a structure, we call it as mechanism schema. Now there are a couple of properties about this schema. Every component that you see in the schema needs an evidence tagging in form of either PubMed ID or bioRxiv link. This can represent uncertainty, and the uncertainty is color-based, where green represents high confidence, red represents low confidence. It can represent ambiguity. You can see the branches here, which can talk about the ambiguous thing that is going on in the mechanism. And incompleteness, which is represented by black oval, when you don't know what is going on, so you can represent what you don't know. So keeping that in mind, we have come up with very general notations to represent the components. So this is notation for substrate perturbation, that is SSP. It's a 3-layered box. The first layer talks about stage at which the SSP is occurring. and which can be from DNA to phenotype, DNA, RNA, protein complex, cell, tissue, organ. And SSP class is an ontology-informed SSP classes, and they are stage-dependent. For example, if you're working with the DNA stage, then the SSP could be a SNV or a copy number, or if you're working with RNA stage, it could be mRNA abundance or edited RNA abundance. These kind of oval represents mechanism module. For representing the unknown, we have the black ovals, so you can use it whatever way you want to when you're building these structures. You can represent environmental factors by this kind of a cloud icon, and you can represent therapeutic intervention sites by the blue octagon. Now, we have developed a web interface which looks something like this, and there is a whole framework behind how will you build this kind of structures. So you can build it, you can version it, you can edit it, you can share it with your collaborators who are part of MecCog system, and you can publish it for a public viewing. Just imagine like you're writing a blog, basically, right? So that's kind of the model that this kind of system is built in. And there are a couple of sections here in this. So this is the drawing place. So one of the component here is a mechanism component section, which kind of catalogs all the mechanism components which you need for your drawing. There's a drawing board here where you can drag and drop your components here, and you can start connecting stuff. There is an annotation form section here for every component that you have. When you click on it, you can get a form which you need to fill in. And here's a control panel which kind of controls the drawing board. You can zoom in, zoom out stuff, or you can auto layout if the structure is pretty complex. And there's a bird eye view if you want to view a really complex, or if you're designing a really complex schema. And this is all backed up by Node.js, Sails.js, and Rapid.js JavaScript libraries. Okay, so you have a framework, you have a software to do it. Now, how does a mature schema look like? So this is a schema that connects a SNV in MST1 gene to Crohn's disease, where your mutation in the SNV in MST1 is here, this SSP, and the Crohn's disease, increased risk for Crohn's disease is this particular box here. And everything that is in between, you're seeing, is connecting your SNV to the phenotype. I'll not get into the details of it, but there are a couple of features which I kind of want to pitch here. One is this entire structure that you're seeing integrates 16 papers, which talks about purely the mechanism of Crohn's disease. And you can actually access those papers by clicking in this pop-up box. When you click in each of those components, this kind of pop-up box opens, and there's a link to PubMed. You can click that, and then you can go and read the paper. The other interesting thing that you see is you can actually see the perturbation across stages. Let's say you want to know what is happening at the protein stage because of this mutation, where these two boxes talks about, okay, this is what is happening in the protein stage, what is happening in the complex stage, what is happening in the cell stage, what is happening in the tissue stage, and finally the phenotype. So you can actually see at the systems level what is happening. You see these black ovals, which represents that you don't know what mechanism is driving this bit of the schema. So this kind of prioritizes what experiments you should conduct in order to transform them. You are seeing branches here, which represents alternative hypothesis that is happening. For instance, here there was one paper that was talking about that because of this SNV, it's weakening the protein-protein interaction, which is represented by the upper part of the schema, and the lower part of the schema represents another work which says that because of the SNV, there was a decrease in the abundance of this particular protein, which impacts the abundance of the complex. So these kind of alternative hypotheses which are there in literature, you can represent that. And the entire structure is saved in form of JSON file. So there's a REST API. You can actually pull this entire structure, and you can do any kind of computational analysis on all the data that is present here. We think of a couple of applications for this, which we are working on. So you can start thinking about, can we build disease taxonomy based on mechanism versus the usual etiology or symptom based? Can we identify therapeutic intervention sites, which we already do using the blue octagon? Can we study epistatic interactions between two genes or two variants? And can we apply this to a precision medicine by understanding what are the possible drugs that can overlap with these kind of structures. So with that, I would like to thank my advisor, Professor John Malt, for guiding me throughout the project, Professor Linley Darden, who is part of the audience here, for making us understand the framework and help us extend the framework. Lipika Ray, she has been, so all the structures, the actual schema structure that you saw was actually built by her, and if you go to the website, you can see many other structures, so she's working on building those structures. And Iju is a postdoc in a lab who is helping with the technical thing. And I would like to thank NIH for funding this thing, this research, and a combined program, which is an NSF-funded network biology program in University of Maryland. And so they have funded my travel fellowship. And I have a poster, and I would be happy to give a live demo, and I would be happy to take any questions.
Well, no, I'm either way.
Yeah.
So can you, what you're doing is modifying the or representing the perturbations from normal, but in order to really understand for a human being what's going on, you'd like some connection to the normal stuff, so I can understand what the differences were, say, in the perturbation. A traditional way to do that might be linking it up through open biomedical ontologies or other kind of controlled vocabularies. Do you have plans for doing that kind of stuff?
Right. So here was a slide. So if you see, where is it? So if you look here in the structure which represents an SSP, the SSP class is ontology-informed SSP class. So we have curated gene ontology, mesh, NCI thesaurus, to look for terms which will fit in here. And in our software, we have embedded it as a drop-down list. And if you don't find those terms, then you can enter a new term, which will be maybe curated later or maybe suggested for a new addition to the ontology. And and often these terms are normal terms. And so we have modifiers for those terms, which like prefixes those ontology terms, like increase, decrease, alters, so that kind of stuff. That's it.